Fine, okay, thank you.
Yes, thank you.
Okay, so the good evening everyone to Professor Kulkarni and also to the organizers, Professor Anand, Shiva and James for your kind invitation and also to arrange the meeting in such a difficult time. So the title of my talk, as you can see, atom counting in zinc oxide by atomic resolution of axis and inline electron holography. So this is a work we published in Journal of Applied Physics last year. So these are actually two advanced microscopy techniques which we use generally to answer very specific materials questions. So I prepared kind of broad general overview on the two techniques. So I'll start with the HRTM or phase contrast microscopy and then introduction of the off-axis electron holography. Then I'll go through concept of resolution HRTM. Then what does it mean by quantitative imaging, different approaches and the results published by different groups. Then our small contribution in the area in terms of your atom counting in zinc oxide by two different your aberration pattern microscopy techniques, and lastly conclusion. So phase contrast microscopy, you all I'm sure aware, was discovered by Janicke in optical microscopy and also awarded Nobel Prize in Physics in 1953. So to understand phase contrast microscopy, you see there's a comparison of the tissue sample without any chemicals staining, right field and the phase contrast image. Here, if you can see the cursor, so you can see if you use a phase plate to do the phase contrast imaging of the weakly scattering tissue sample, you can see lots more details which are not available in the bright field image. So you can understand how phase contrast work with the help of this of the phasor diagram. So you know the wave of the vibration MV and MV prime are represented by using the complex diagram, and the horizontal component MP, MP prime are the amplitude, and the angle from the horizontal component is the phase. So if you have a strongly scattering sample, then illumination MA changes from MA to MD prime, and the compounding MP is MD. So the feature appear dark in a white background. But if you have a weakly scattering sample, so if you don't have any chemical staining, then you don't change the amplitude, you only change the phase. So amplitude changes from MA to MD. So the compounding image is MD prime. So to be able to see this effect, you need to rotate the phase of either MA or MD to MPH to be able to see the contrast. And that is performed actually by using a phase plate. The phase contrast microscopy, can you hear me? Hello, can you hear me?
Yes, sir. Please go on.
Okay, thank you. So phase contrast microscopy in HRTM means the periodic dots, and these periodic dots actually represent the lattice structures of the crystals along high symmetry orientation. And here are some examples from our own work: nickel ferrite along 1G orientation showing the spinel structures and A-side cation defects. 2D material MOS2 grown on C-plane surface cross-sectional image showing the stacking and the interfacial binding. MOS2 RES2 alloy showing the phase transition between 2H and 1T. So this is also another technique called atomic resolution jet contrast technique, which also can give you the similar kind of dots, like HRTM. What we added information, but you also get the chemical contrast. For example, silicon and nitrogen to graphene, you can see the brighter dot has silicon and the nitrogen. So you have chemical contrast. So just when I'm showing this image, actually, I should mention that your HR team is also able to give you this kind of information, but not widely used. Slide on inline and off-axis holography introduction. Holography was proposed by Denis Gabor in 1948 and also awarded Nobel Prize in Physics in 1971. So holography means the complete information recording of the wave field. So the purpose of Gabor was basically to get rid of the aberration from the electron micrograph, because at that time the quality of the instrument was very poor. So it's a two-step process, writing and reading. So Gabor's proposal was inline holography, and he had to abandon his method because he could not get rid of the twin image issue. Until Leith and Upatnaik used a tilted beam illumination, which separated the DC component and also the twin image components. And holography is a very well-established and a widely used technique in many branches of science and technology. But to do the holography with the electron, you need electron biprism, and the electron biprism was discovered by Gottfried Mollenstedt, and this is one of the first electron interference fringes. And here is a schematic geometry of the electron interference fringe under off-axis geometry. So basically, in the off-axis geometry, you add a carrier frequency, and the intensity pattern is given by this expression, and Q is a carrier frequency of the hologram. And here is the basic schematic of the light optical analog of the electron biprism, which is known as the Fresnel biprism. This slide showing the schematic principle behind HFTM and the off-axis electron holography. So for the HFTM or the inline holography, the defocus HFTM actually is equivalent to inline hologram. So you basically have to collect lots of images, at different focus. And for holography, you have a biprism. But to work at atomic resolution, you need double biprism because the field of width is very small at atomic resolution, and the microgravs suffer from the Fresnel fringes and also vigneting effect. And the double biprism setup actually to help to get rid of these effects to give you better micrograph and to extract better numbers. So at the end, what you are interested actually to recover the object XZ function of the form psi equal to a to the power I phi. So the holography at atomic resolution was performed in the tube Berlin microscope. This is a special microscope, aberration corrected, and also equipped with the double biprism. And the HFTM we have recorded in our own microscope in JNCSR. Both are aberration corrected, both are spatial resolution better than 0.1 angstrom, 8 angstrom, and then we have variable CS. Holography at medium resolution is a well-established technique and to image electromagnetic fields in materials. And here are some examples from literature. For example, doping profile in the NMOS transistor, then vortex lattice in ion superconducting film, also ferroelectric polarization boundaries and domain in the barium titanate. And also holography has been used to image the flux closure and also remnant field in the cobalt nanoparticle and also to image the scannion lattices. At atomic resolution, you have to record hologram at the sub-angstrom length scale. There's an example of silicon along 110 orientation showing the lattice of silicon, and in that you can see the fringe is at the sub-angstrom length scale. And here on the right hand side is the amplitude and the phase extracted from such atomic resolution hologram of the gallium arsenide sample along 110 orientation. So what does it mean actually? You have amplitude and phase information from the atomic and the subatomic length scale. So that not only helps to identify and count the atoms from the individual dots, but also you have information from in between the columns. And the in between column information is very, very important because you have electrons residing there and you have a lot of chemistry going on there. So now coming to the microscopy and the concept of resolution. You all know that to be able to distinguish, we define resolution by to be able to distinguish or discern between 2 points. And you all know the human eye can resolve max 100 to 200 micrometer. And optical microscope is used to be able to see your lens colors feature below 100 to 200 micrometer. And if you have ideal lens, then you image point. You image a point as a disk because your diffraction limited, and this disk is known as AD pattern, and the expression for the AD pattern is gained by 0.61 lambda over mu sine beta, where lambda is the wavelength of the illumination and mu sine beta is the numerical aperture. And the Radius criteria of resolution is defined by a distance d naught. So it's not a very strict criteria. So it's typically defined by the distance between two such AD pattern, where the minimum of the first pattern falls right below the maximum of the second pattern. And the d naught for optical microscope is gained by this particular equation. So aberration. So in the electron microscope we have aberration. So your we have severe blurring from the aberration. There are the two examples of spherical aberration and the chromatic aberration. So you can see the point object is blurred in the Gaussian image plane significantly, and the spherical aberration is because of the inhomogeneity of the lens, and the chromatic aberration is because of the spread of the lambda in the illumination. But there is a plane called Sarger plane, which can be accessed by applying a little bit of focus. And this plane is known, or the image is known as disk of least confusion. So in the presence of the aberration, but mentioned that we don't have Janicki-like display in the electron microscope. So we make use of this combination of the CS and T focus to basically apply phase contrast. And in the presence of aberration, we define resolution by using the PCTF function. It's known as phase contrast transfer function. So the first zero crossing of the PCTF will give the point resolution GS, and GI is the information limit. So the information between GS and GI is not straight. It cannot be accessed correctly. And the ideal shape of the PCTF should be like this to minimize the gradient of the PCTF function, to minimize the localization in the image. If the environment is not very stable, if the noises coming from the environment, for example, stray electromagnetic field, vibration, and all, they appear as incoherent aberration, and they basically lower the point resolution of the system. So in the aberration corrector microscope, now we have Rose corrector. And this was developed by these three professors, Harold Rose, and the Haider, and then Urban, and also awarded full prize in physics in the year 2011. And so the principle behind of aberration corrector is that you don't make lenses aberration free, the principle is that geometrical aberration adds up. So you have to nullify the effect of one lens by another set of lenses. So we have double hexapole and the two transfer doublet. This is for the imaging system, for the broad beam illumination, and the bottom one for the probe corrector system. So same mode. So I'll not go through the details of that because there is a long history and struggle in this field. So here's a brief comparison between the off-axis and the inline holography of the HRTM. So your HRTM basically acts as a bandpass filter. So it only transfers information with the high contrast only for specific frequency range, and that we basically align it to the interatomic spacing. And for holography, actually can transfer all frequency range information. To understand this, you can take this example. Is a photograph of the Goldfried Molenstret. You can say he's doing something, and if you do the Fourier transform of the image and then filter out some of the frequencies and then back Fourier transform to form the image back again, you can see that you can still make what person is there. He's doing something, but you lose a lot of information. For example, the contrast. So the contrast difference, so the shade difference between the hair and the wall shot and the pole. So your HRTM actually is something like this, but it's like this. Actually, it's slightly based on present methodologies. So now there are examples in the literatures. So you can do the quantitative imaging following different route. You can follow holographic approach. You can follow HRTM approach, and also there are other methods like electron probability current. Some examples from literature. Here is an example of nitrogen doped graphene. So it is not only the imaging of the atomic dot, but it's showing actually how one electron of nitrogen is shared by surrounding carbon atom. So one electron wave function actually is imaged by this technique. And from one micrograph, the complete reconstruction of all the atoms in the MgF crystal has been shown, and also for the bilayer graphing by another theoretical method called Big Bang tomography. So, what is the eventual goal of the quantitative imaging? Can we determine materials properties through imaging? And if you want to do that, then what does it take? Because materials have atoms, they have particular arrangements, and the materials can have defects, dopants, etc. And if you have atoms, then you have electrons around it, and you have electrons, you have field around it, potential around it, and if you can determine the potential, and once you can fit the orbital, you should be able to basically tell the properties of the material in terms of magnetic moments and other things that you're interested. So there is no restriction or limit posed on the theory. And it's quite possible because nowadays tremendous advancement has been made, both in the field of microscopy and also in the field of detectors. So I'm sure that people can take up this kind of difficult challenges. Now coming to the result part, so we have studied zinc oxide thin film. epitaxially grown on zinc oxide substrate, known as C-plane of the substrate. And our initial goal actually was to understand the native defects by imaging between the N-type and the R-type sample, where we have carrier difference 10 to the 19 in one case and 10 to the 16 in another case. But due to some limitation at that point of time, we restricted ourselves only for the atom counting. So experimentally for HRTM, you have to collect your series of images at different defocus. And if the sample is thin, then you are within the linear imaging condition, and there exists the analytical solution within the weak phase object approximation. But if the sample is thick, then you are dynamical scattering, then you are within the non-linear imaging condition, and you have to follow certain iterative approaches. For example, PAM, MAL, to retrieve the amplitude and the phase of the object executed function. In case of holography, it's quite straightforward. Once you do the Fourier transform of the hologram, you have three bands. You have central band and the two side band, and two side band are the twin image to each other. So you can see that you can separate them out in the frequency space. Now you take one of the side band out and do inverse Fourier transform, and you have the complete wave function information. It's not straightforward, will come, but you have to fit to the model calculation. Here is a schematic flow of the data analysis. I already said. So for the off-axis, you do the FFT, take on the side band, do inverse FFT, get the complex image, and get the phase and amplitude out. And for HRTM, you collect through focal series, and you follow certain iterative. If the sample is thick, algorithm, then you get object exit wave and get the phase and amplitude. So there is one important parameter called phase detection limit. So it's like resolution. Phase detection limit also is very, very important because it will tell you what is the smallest gradient of the electromagnetic field you can determine. So I'll not go through the details of it, but from our experiment, we have phase detection limit about 0.007 rad, which is actually good, much better than the requirement, because to count incremental oxygen and the zinc on the column, you need 0.1 and 0.2 rad. So the numbers we have actually will allow you to count the oxygen and zinc atoms. So in this context, the other report reported 2.5 over 80, 2.5, tell me?
So you have 1 more minute?
Sure, I'll finish. I'll not take not much, another few minutes maximum.
Sure.
So Cooper et al. They reported 2 pi by 1000. So, but none of them actually have used the double prism or elaboration microscope, and the numbers also not better than us. So, and the phase detection limit in case of HRTM not discussed, except the Izima's paper, the peak phase value of nitrogen boron 0.0 to 0.022. Dimension and the difference of 0.004. Anyway, I'll not go through much on that. And you also need to model the system, how electron and the wave and the matter interact, and how it get basically further change due to the lens parameter or the lens aberration, to calculate the intensity, the phase and amplitude eventually. So I'll not go through the details, but we have followed the Hartree-Fock potential, and we calculated intensity by using this particular equation from certain source, and we calculate the positive potential using the Hartree-Fock as we mentioned already, and then calculated phase by this equation. And also we took care of the inner bond and the outer bond with potential, because potential is asymptotic function. We found that beyond certain inner bond and beyond certain outer bond, the atomic scattering factor and the intensity and phase do not change significantly. But experimentally, you are restricted on the inner bond and the outer bond because of the limitation on the interatomic spacing. So for zinc and oxygen, they are closely situated to each other. So we have selected 1 to 50 and 1 to 25 for zinc and oxygen for the mean phase. So you can basically consider two different phase value, peak phase and mean phase. Anyway, so now, so this is the result. You can see the reconstructed wave function both from the central band and side band. So central band, again, I forgot to mention, is equivalent to the HRTM or the Indian holography from the off-axis holographic data. And side band actually is the off-axis hologram. So we have the amplitude and phase, and the arrows are showing along which we have counted, we have evaluated the phase and the corresponding atom number. And zoomed in image showing the red one is the zinc atom and the yellow one is the oxygen atom. So is a summary plot showing central band, side band, your comparison. For very thin area of the sample, you see the systematic increase both in the phase value and the atom number with the sample thickness. And the nice thing is that zinc and oxygen, though the contrast is different, and the atomic number difference is very high, but in the neighboring column, we have obtained the same number. This is a very nice result, and the methods that people have used. But for thicker area, the trend is not very good with the sample thickness. That may be because of dynamical effect and also how we basically handle the data. But we don't have very good result actually from the HRT image series and our and a commercial MacCampus code that employs the Gauss-Saxon algorithm, particular algorithm. It doesn't give any systematic trend. So thin and thick, respective thin and thick area, we obtain almost the same number error. Anyway, so now coming to conclusion, because I just try to make it simple and then to the briefly. So, but the major conclusions were for thin sample area and the center band side band reconstruction yield good agreement. However, for thick sample area, agreement no longer holds. And 3 to 4 times more atom numbers is obtained considering mean phase value compared to the peak phase value. HRTM reconstruction gave almost constant atom number, one irrespective of sample thickness, and both HRTM and the off-axis holography at atomic resolution offer opportunity both in material study, method development, and also fundamental science from subatomic scale. Acknowledgement, a special thank to Professor Sienna Rao, founder president, for the advanced electron microscopy facility and also giving me the opportunity to work in such a microscope. And sincerely thank ICM and CPM for research funding. A special thank to DFG for the work in Q Berlin. And lastly, to all of my students for all the work, and then last to all of you, for your kind attention. Thank you.
Thank you, Dr. Ranjan, for giving such a nice talk. We have time for only one question, or maybe
Sure.
offline.
Sure.
Any questions?
Sure.